Hello everyone, my name is Jeannette Bouchard and I am an infectious diseases pharmacist at WakeMed Health and Hospital System. In this presentation, I will be reviewing the pertinent drug information regarding the SARS-CoV-2 mRNA vaccines. In this video, we will be discussing the SARS-CoV-2 vaccines utilizing mRNA technology. As of December 26, 2020, there are 22 vaccines utilizing RNA technology in preclinical and 7 in clinical development. 2 of these vaccines are currently EUA approved in the United States, BNT162b2 from Pfizer-BioNTech and mRNA-1273 from Moderna. These will be the main focus of the video. As other mRNA vaccines are approved or have data published, this video will be updated to reflect data surrounding those vaccine candidates as well. Pfizer's BNT162b2 is currently EUA approved as a 2-dose regimen 21 days apart. The storage requirements are -70 degrees Celsius, and further details regarding storage will be discussed later. And the approximate cost of this vaccine is about $20 per dose. Moderna's mRNA-1273 is currently EUA approved as a 2-dose regimen 28 days apart with storage requirements of -20 degrees Celsius and an approximate cost of $32 to $37 per dose. mRNA technology was first reported in 1990 utilizing reporter gene injections into mice to create proteins. Only a few years later, this technology began to be advocated for as a vaccine platform. Interest in vaccine potential peaked because of mRNA's non-replicative nature, lack of genomic integration, and general instability. These characteristics made it the perfect messenger to carry important information when compared to DNA and viral vector technology. The testing of the first mRNA vaccine was in 1993. The mechanism of mRNA vaccines is depicted in the figure on the slide. RNA encoding the viral spike protein is encapsulated in a lipid coat or lipid nanoparticle. This lipid nanoparticle allows the RNA to enter the cell where it is translated in the ribosome to protein. This protein is then presented on the cell surface to be recognized by CD8 T cells, generating cell-mediated immunity. These protein particles may also be transported outside the cell where they can be identified by other immune cells in the body and create antigen-specific antibodies. There are 2 classes of mRNAs commonly used as vaccine vectors, non-replicating and self-amplifying mRNA. Both types are currently being studied for prophylactic vaccines for infectious diseases. Concerns with this technology were threefold. 1. mRNA is incredibly unstable and prone to degradation by RNases, which hindered its delivery to cells. Research dealing with delivery systems for mRNA have been a large focus for this technology and involves the use of cationic molecules, lipids, polymers, and nanoparticles. 2. mRNA is highly immune activating since it is recognized by a variety of cell surface and innate immune receptors, a feature of mRNA that could be incredibly beneficial or just as equally detrimental. And 3, it's inefficient in vivo delivery, which has since been improved with the current vaccines by utilizing a lipid-based vector. Other technologies such as microinjections and RNA patches have also been used, but not in the context of the currently studied SARS-CoV-2 vaccines. The first reported use of these lipid-based vectors was in 2015. These lipid-based vectors are internalized and attached to the cell membrane via their non-bilayer lipid phases. As technology has progressed since 1990, many of the original concerns regarding mRNA have been addressed, and more stable and highly translatable products are available and being tested. As of 2020, there were at least 6 phase 1 or 2 clinical trials ongoing for vaccines against infectious diseases such as CMV, Zika virus, and rabies. First, we will review Pfizer-BioNTech's BNT162b1 and b2 candidates. Non-clinical data for BNT162b2 is summarized in the table shown. It is important to keep in mind when reviewing immunogenicity data for these vaccine candidates that no correlative protection for SARS-CoV-2 at the time of this video has been established. However, based on previous data regarding protection against viruses in humans, and animal rechallenge data such as what is showcased on this slide, neutralizing antibody levels may correlate to protection, but this is not definitively known. In 2 separate rat models, once-weekly dosing for a total of 3 weekly cycles of 30 microgram and 100 microgram BNT162b2 doses showed no systemic toxicity and a robust immune response was identified. A single injection elicited high neutralizing titers and a Th1 T cell response. Th1 T cells produce interferon gamma, tumor necrosis factor, IL-2, and are mainly involved in the protection against intracellular pathogens. Th1 dominant T cell responses were associated with less severe cases of SARS-CoV-1 when compared to Th2 T cell responses, which were associated with enhancement of lung disease following infection. This data has been extrapolated to SARS-CoV-2 to avoid immune responses that could potentially enhance disease. In a non-human primate immunogenicity study, currently only available as a preprint, the immunogenicity of BNT162b2 in macaques paralleled that previously seen in mice. 7 days after dose 2 showed a geometric mean concentration of the S1 binding IgG in the 30 microgram dose that was substantially higher than convalescent sera. 50% geometric mean titers of SARS-CoV-2 neutralization assays peaked at days 14 after dose 2 in the 30 microgram dose, and levels above 285 persisted for at least until day 56 or 35 days post-dose 2. During re-challenge of SARS-CoV-2 in the macaques, no virus was detected in BAL fluid through day 6, which when compared to control immunized macaques was statistically significant. In regard to nasal swabs, detection of virus in the immunized group only occurred on day 1 post-challenge and not on days 3 or 6. Currently, a developmental and reproductive toxicity study is ongoing for BNT162b2, but this data has not been released or published at the time of this video. This promising animal data led to the continuation of phase 1 and 2 dosing and safety trials. There were 2 primary phase 1 studies for Pfizer's BNT162b vaccines. Notably, during the preclinical and phase 1 data, there were 4 potential Pfizer vaccines, which following clinical trial results in Germany, this 4 was narrowed to 2 potential candidates, which were then tested in the United States. Ultimately, it was decided to only move forward with BNT162b2 for the full phase 3 trial, given the information derived from the phase 1 and 2 data, which will be discussed over the next few slides. BNT162b1 encodes for the receptor binding domain of the SARS-CoV-2 spike protein, and BNT162b2 encodes prefusion membrane-anchored SARS-CoV-2 full-length spike protein. During this review, we will focus specifically on the data that compares the 2 vaccine candidates from Walsh and colleagues, as this directly speaks to the ultimate vaccine candidate, BNT162b2. Groups of participants in the trial were to receive doses of 10, 20, or 30 microgram doses of either vaccine candidate or placebo on a 2-dose schedule 21 days apart. Groups were also broken down by age, with one group including ages 18 to 55 years and the other ages 65 to 85 years. The primary safety endpoints were solicited local reactions and systemic reactions, and the use of antipyretic or pain medication prompted by and recorded in an e-diary. Immunogenicity assessments included SARS-CoV-2 neutralization assay and receptor binding domain or S1 binding IgG immunoassays performed at baseline, 7 and 21 days after the first dose, and 7 and 14 days after the second dose. Human convalescent sera was included as a benchmark for immunogenicity data. The serologic responses were similar between candidates, and because BNT162b2 is the candidate that fully progresses to phase 3, its neutralizing titers are showcased here. In the figure shown, by day 28, or 7 days after a second dose, in both the younger and older age groups, 30 micrograms of BNT162b2 elicited a neutralizing titer response equal to or above the human convalescent sera. Consistent with the data seen in non-human primates, the highest neutralization titers were seen at 7 and 14 days after the second dose. Of note, for both candidates, there was an overall decrease in antigen-binding IgG neutralization titers seen in the older age group. This effect is similar to what is observed in vaccines for other diseases. The safety data regarding these 2 candidates is where their differences lie. For BNT162b1, the predominant local reaction reported was pain at injection site, with higher doses eliciting higher local reaction reporting. Local reactions were increased after dose 2 and were reported in similar proportions in both the older, which is not pictured, and younger age groups. Local reactions reported for BNT162b2 were similar to B1. However, there were less reported redness and swelling in the younger age group and no reports in the older age group. There were no grade 4 local reactions reported for either vaccine candidate. The systemic events for BNT162b1 were predominantly fatigue. However, there was a substantial number of participants who reported fever and chills, specifically after dose 2 and in the 30 microgram dose group. Systemic events were less in the older patients when compared to the younger patients. These systemic events peaked by day 2 and resolved by day 7. Systemic events reported for BNT162b2 were lower when compared to B1, as shown on this slide. Following dose 2 of the 30 microgram dose for this candidate, the percent of participants who reported any fever was 17%, compared to 75% reported for the 30 microgram dose of BNT162b1. This pattern was consistent for the older age group as well, which is not shown on this slide. Additionally, after the first dose of BNT162b2, systemic events occurred at similar rates to placebo, especially in the lower dose groups. Based on the data presented in the U.S. Phase 1 trial, it was decided to move forward with the best candidate, BNT162b2. This was based mostly on the safety data differences, specifically the systemic reactions, as both candidates produced similar immunogenicity data. Next, we will discuss the pivotal efficacy trial for BNT162b2. CS4591001 is an ongoing Phase 3 placebo-controlled, observer-blinded efficacy trial for participants ages 16 and older, an amendment that was added in September for the U.S. specifically. This trial evaluates the current data on safety, immunogenicity, and efficacy of BNT162b2. Data regarding durability of immune responses and immunogenicity is ongoing and not included in the published dataset. Participants included were healthy or had stable chronic medical conditions, including HIV, hepatitis B, and hepatitis C. Patients were excluded if they had a medical history of COVID-19, were immunocompromised, or were on immunosuppressive therapy, were pregnant, or were breastfeeding. The primary efficacy endpoints were confirmed COVID-19, defined as a positive SARS-CoV-2 nucleic acid amplification-based test with one or more mild COVID-19 symptom, with onset greater than or equal to 7 days after the second dose, in patients without serologic evidence of SARS-CoV-2 at randomization. A second primary endpoint included this outcome in patients both with and without serologic SARS-CoV-2 evidence at randomization. Secondary efficacy endpoints included efficacy against severe COVID-19, defined as having serologic evidence of SARS-CoV-2 with clinical signs indicative of severe systemic illness such as respiratory failure, ICU admission, or death. The primary safety endpoints of the trial were solicited local and systemic adverse events, the use of antipyretic pain medications, and unsolicited adverse events. The primary safety endpoints were broken down into various timeframes and denominator values. Solicited local and systemic events and the use of pain medication were recorded within 7 days of receipt of vaccine and constituted the reactogenicity population. By design, this had to include at least 6,000 participants, and at the time of publication included a total of 8,183 participants. Unsolicited adverse events were reported through 1 month after the second dose, and unsolicited serious adverse events were followed up to 6 months after the second dose. Adverse event data through about 14 weeks after dose 2 was included in this report. Safety analyses for participants infected with HIV were not included here. 44,820 participants were screened and 43,548 were randomized at 152 sites worldwide, including the United States, Argentina, Brazil, South Africa, Germany, and Turkey. 43,448 participants received injections and 37,706 had 2 months of available data as of October 9th. The baseline characteristics for this group are shown here. 50.6% were male, 82.9% white, with the majority, about 57.8%, falling into the younger age group. Notably, the participants ages 16 to 17 had a limited representation of 153 at the time of this report. Despite efforts to ensure diversity by enrolling participants on a global scale, 76.7% were enrolled in the United States, with Argentina having the second most participants enrolled at 15.3%. 35% of participants had a BMI greater than or equal to 30, and 20.5% had a baseline comorbid condition classified according to the Charlson Comorbidity Index. Of note, HIV patients were included in the safety population. However, their safety data did not contribute to the overall analysis of this population at the time. Overall baseline characteristics in the 2-month follow-up population were similar between treatment and placebo arms. Of the approximate 36,000 participants who had no evidence of infection prior to enrollment, there were 8 in the treatment arm and 162 in the placebo arm who were observed to have symptomatic COVID-19 at least 7 days after the second dose. This equates to a vaccine efficacy of 95%. For the secondary primary efficacy endpoint, in those patients with and without prior infection at enrollment, there were 9 and 169 cases in the treatment and placebo arms respectively, which equates to a 94.6% vaccine efficacy, demonstrating that the true vaccine efficacy in the population most consistent with the real-world scenario is at least 89.2% with the current data. For the secondary efficacy endpoint of prevention of severe COVID-19 disease, there was 1 case reported after 1 dose in the treatment arm and 9 in the placebo arm in the modified intention to treat population, which equates to a vaccine efficacy of 88.9%. When looking at severe COVID-19 cases occurring at least 7 days after dose 2, there was 1 case in the treatment arm and 4 in the placebo arm, which equated to a vaccine efficacy of about 75%. Due to the small number of severe COVID-19 cases available, the posterior probability that the vaccine efficacy for this endpoint was greater than 30% did not meet the pre-specified criteria of greater than 98.6%, and these results should be interpreted cautiously until more data is available. The efficacy between the first and second doses was 52%, which suggests that there may be some protection following the first dose, but for optimal protection, both doses need to be administered. This study was not designed to evaluate the efficacy of following 1 dose, and thus the follow-up timeframe for this data is short. When subgroups were analyzed, outcomes were generally consistent with the overall population, including those with hypertension and in high-risk groups such as older age and obesity. Although it is important to note that this study was not powered to assess efficacy in these subgroups. When looking at the local reactogenicity population, there were more local reactions in the treatment group than the placebo group. However, most were mild to moderate in nature. Severe local reactions had a frequency of less than or equal to 0.6%, with no grade 4 or potentially life-threatening reactions reported. Injection site pain was the most frequently reported local symptom, and this was also reported more frequently in the younger age group than the older age group, as shown in the graphs. The younger population is located at the top and the older population is the bottom. Overall, local reactions were similar after dose 1 when compared to dose 2, with redness and swelling being the most consistent. Most reactions resolved within a few days of onset and were largely driven by injection site pain. Systemic reactogenicity outcomes again were higher in the treatment group than in the placebo group, with the exception of vomiting and diarrhea, which was similar to placebo. Certain reactions had increased frequency after dose 2, such as fever and muscle pain. Overall, the most noted systemic reactions were fatigue and headache. Again, older patients seem to be less likely to have systemic reactions than younger patients. After both doses, the use of antipyretic or pain medication was slightly lower in the older group compared to the younger group, and trending similarly with fever, which increased after dose 2. Most of the systemic events were mild to moderate in severity. Severe systemic reactions were rare and occurred in less than 2% of patients. It is important to note that systemic reactions were observed within a median onset of 1 to 2 days following vaccination and resolved within a median duration of 1 day for both younger and older age groups. For overall adverse events, as mentioned earlier, there were data available for 2 months post-dose 2 in 37,706 participants, but results for the total enrolled population are shown here. There was an increased adverse event rate in the BNT162b2 arm versus the placebo. However, when looking at serious adverse events or events leading to study withdrawal, BNT162b2 was comparable with the placebo, with rates being less than 1%. There were no clinically meaningful differences in adverse events when broken down by subgroups of age, sex, race, or baseline SARS-CoV-2 status. When broken down by the group that had 2 months of follow-up following dose 2, rates of adverse and serious adverse events were similar. There were 64 reports of lymphadenopathy in the treatment arm compared to 6 in the placebo arm, with the majority of these being considered related to study intervention. These were reported within 2 to 4 days of vaccination and continued for an average duration of 10 days. Of the 6 reported deaths in the study, 2 in the BNT162b2 arm and 4 in the placebo arm. None of them were related to vaccine, placebo, or COVID-19. It is important to note that there were 4 reports of Bell's palsy in the BNT162b2 arm and 0 in the placebo. Although these rates are consistent with the expected background rate, it is an area noted to be watched closely in the future, due to the numerical imbalance between groups. 1 case of Bell's palsy was noted to have resolved at the time of data cutoff, and the other 3 were reported as continuing to resolve. In summary, based on current data from both preclinical and clinical trial data, BNT162b2 is safe and effective against COVID-19 with 95% efficacy. Serious adverse events were low and consistent between treatment and placebo arms, including in patients who are SARS-CoV-2 serologic positive and asymptomatic at baseline. It is important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces. Encouraging vaccination at the end of the work week may be appropriate to avoid overlap. This large phase 3 study not only delivered positive results and was produced with large numbers in a short period of time, but it also provides proof of concept for RNA-based protection against infectious diseases, which will be huge for the future of this field. In terms of what is missing from the current data, there are a few gaps that these leave us with. The major gaps for the general population are the long-term safety and efficacy data, as well as the vaccine's efficacy against asymptomatic disease and protection against severe disease. This study was not designed to evaluate efficacy against asymptomatic disease and disease transmission. There were also too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease. Further data will be needed to address these issues, and until then, continued public health reminders such as continued mask wearing and social distancing is strongly encouraged. Further data is also needed in specifically excluded populations such as pregnancy and breastfeeding and immunocompromised. There is currently no data available to speak to safety and efficacy in these populations, with the exception of the DART animal study mentioned earlier that we are awaiting results for. As more data from clinical trials and patients receiving the vaccine via EUA access is available, we will hopefully have more information to speak to these populations. Of note, there is a clinical trial starting in patients aged 12 to 16 and preliminary data available in the Pfizer-BioNTech briefing document regarding safety in younger adolescents. However, we currently only have data for 100 participants, and more information will be available in the future. Next, we will review the available data for Moderna's RNA-based vaccine candidate mRNA-1273. Moderna's preclinical work with MERS-CoV and SARS-CoV-1 allowed them to evaluate several candidates for SARS-CoV-2 and quickly determine the most effective design, the spike protein. The evaluation of mRNA-1273 in mice is available via a preprint, and briefly, it showed potent neutralizing antibody and T-cell responses, as well as protection of SARS-CoV-2 infection in lungs and nasal passages of mice without evidence of vaccine-associated lung immunopathology. Immune responses in mice were noted to continue for more than 2 months following similar regimens currently being utilized in human trials. In non-human primate studies, data for which is shown on this slide, macaques were vaccinated at week 0 and 4 with either 10 or 100 microgram doses of mRNA-1273. At week 8, macaques were challenged with SARS-CoV-2 via intratracheal and intranasal route. There was a dose-dependent response for both IgG binding and neutralizing activity. This peaked at 2 weeks post the second vaccination. The IgG binding and neutralizing geometric mean titers were 5 and 15 times higher than convalescent sera samples 4 weeks after the second dose of the series. Dose-dependent Th1 T cell responses were noted 4 weeks after vaccination in 4 of 8 animals in the 10 microgram dose and 7 animals included in the 100 microgram dose. Th2 T cell responses were low or undetectable in both groups, which is important due to its link to vaccine-associated immunopathology as previously described in BNT162b2 preclinical data. Upon viral rechallenge, 2 days after, only 1 of 8 animals in each dose group had detectable RNA in BAL fluid, compared to 8 of 8 in the placebo group. None of the treatment group animals had detectable RNA in nasal passages, compared to 5 of 8 with placebo. This promising animal data led to the continuation of Phase 1 and 2 dosing and safety trials. The Phase 1 preliminary report for mRNA-1273 was published on July 14, 2020, with additional data on older adults being published originally in September. This was a dose escalation open-label trial of 45 adults 55 years of age or younger and 40 adults 56 years and older. Participants received 2 vaccinations separated by 28 days of 25, 100, or 250 microgram doses. Due to increased reactogenicity without significant immunogenicity, the older age group only received the 25 microgram and 100 microgram doses. Follow-up visits were on days 7 and 14 following each vaccination and on days 57, 119, 209, and 394. Of note, participants were asked to not use antipyretic or pain medications before or after vaccination. Immunogenicity data from this study indicated that a 100 microgram dose as 2 injections 28 days apart resulted in neutralizing antibody induction by day 7 post-dose 2, as seen on this slide. Specifically, in the 100 microgram and 250 microgram dosing groups, by day 7 after dose 2, geometric mean titers for neutralizing titers were similar in magnitude to each other and were comparable to the upper half of the convalescent sera distribution. Again, similar to preclinical data, T cell responses strongly favored the Th1 T cell expression, and there were low-level Th2 T cell expression. In participants who were 56 years or older, similar high-level neutralizing and IgG antibody binding was exhibited to the younger participant group. As shown on the slide, neutralization increased rapidly 7 days after the second vaccination, further emphasizing the importance of the second vaccine in both age groups. This data is based on a smaller number of participants than included in the younger age group, and full immunogenicity data will likely need further elucidation in this older population. Key safety findings from the Phase 1 data in younger participants is shown on this slide. Overall, the most common solicited adverse events were headache, fatigue, myalgia, chills, and injection site pain. There were no serious adverse events reported. After the second dose of the vaccine, local and systemic reactions were more common and more likely to be moderate in severity. The graph shown specifically depicts an increase in fever, chills, and myalgia, and reactions were driven by dose with the 250 microgram dose having the higher frequency of adverse events reported. For the older age group, there were no observed differences seen with the younger age group. These limited differences could be due to the small number of participants included in the Phase 1 study. In addition to continued safety and immunogenicity monitoring with Study 101, the study addressed over the last few slides, Study 201, which is a Phase 2A study addressing 50 and 100 microgram doses in participants ages 18 and above, also looked at safety and immunogenicity in these specific doses. These results have not been published but are available in the Moderna briefing document for EUA approval. The data in this Phase 2A study continued to support the immunogenicity findings of the 100 microgram dose of mRNA-1273 and further solidified it as a top dosing candidate for Phase 3 clinical trials. Next, we will discuss the pivotal efficacy data for mRNA-1273. Study 301 is an ongoing Phase 3 placebo-controlled efficacy trial for participants aged 18 and older. This trial evaluates the current data on safety, immunogenicity, and efficacy of mRNA-1273. Of note, this data is from the available data that was last updated on November 25, 2020 from Moderna's EUA approval briefing document. Participants included were healthy or had stable chronic medical conditions such as HIV, hepatitis B, and hepatitis C. Patients were excluded if they had a medical history of COVID-19, were immunocompromised or on immunosuppressive therapy, were pregnant, or were breastfeeding. They were randomized in a 1-to-1 ratio that was stratified based on age and presence or absence of risk factors for severe illness of COVID-19. Treatment groups included 2 100 microgram doses of mRNA 1273 28 days apart and 2 doses of placebo 28 days apart. The primary efficacy endpoints were confirmed COVID-19 that was defined as a positive SARS-CoV-2 test with 2 systemic or 1 respiratory COVID-19 symptom, with onset greater than or equal to 14 days after the second dose in patients without serologic SARS-CoV-2 at randomization. Secondary efficacy endpoints included efficacy against severe COVID-19, which was defined as having SARS-CoV-2 with clinical signs indicative of severe systemic illness such as respiratory failure, ICU admission, or death, COVID-19 regardless of symptomatology or severity starting 14 days after the second injection, and symptomatic COVID-19 based on a less restrictive criteria of 1 or more symptom. The primary safety endpoints of the trial were solicited local and systemic adverse events, the use of antipyretic pain medications, and unsolicited adverse events. All participants assessed provided a nasopharyngeal swab and blood sample for SARS-CoV-2 before the first and second injection during the study. 3,381 participants were randomized. Overall, 52.6% were male and 79% white, with the majority of participants being between the ages of 18 and 65. The study had planned to enroll at least 25% and up to 50% of participants most at risk for severe complications, which based on the full analysis report on November 25th was achieved with 41% being above 65 years old or younger than 65 with a severe risk factor such as diabetes, severe obesity, or chronic pulmonary disease. As with the clinical trials for BNT162b2, participants included were at high risk for disease acquisition based on their occupation. In Study 301, about 25% of participants were healthcare workers, with the remainder being other frontline workers that would put them at risk of disease acquisition. Baseline characteristics between groups appear to have been well balanced between treatment and placebo arms based on the available data. For the primary endpoint of symptomatic COVID-19, at least 14 days following the second injection in baseline seronegative patients, there were 11 cases in the treatment arm and 185 cases in the placebo arm, correlating to a vaccine efficacy of 94.1%. The slide shows the interim dataset cumulative incidence rate of time to first occurrence of COVID-19 starting 14 days after the second injection as of November 25th. With the red line representing mRNA 1273 and the blue line representing placebo. For symptomatic COVID-19, regardless of prior SARS-CoV-2 infection, there were 12 cases in the treatment arm and 187 cases in the placebo arm, with a vaccine efficacy of 93.6%, which is consistent with the primary outcome, but is more representative of the real-world population. Since only one participant who was previously COVID-19 positive had symptomatic COVID-19 disease, these results should be cautiously interpreted and further evaluation is needed. Regarding secondary endpoints, there were 0 cases of severe COVID-19 in the treatment arm and 30 in the placebo arm, correlating to a point estimate of vaccine efficacy of 100% for preventing severe disease. One of the cases of severe COVID-19 disease unfortunately resulted in death in the placebo arm. In patients who had only received 1 dose of vaccine at the November 11th interim analysis cutoff, vaccine efficacy was 80.2% with a median follow-up time of 28 days. With such a short follow-up period in a trial that was not designed to assess this vaccine after 1 dose, there is not sufficient data to suggest that 1 dose is appropriate and a full 2-dose schedule is needed for optimal protection. When vaccine efficacy was evaluated for various subgroups, efficacy was consistent across major demographic and baseline characteristics and similar to outcomes in the primary efficacy group. There was a numerically lower vaccine efficacy in higher age groups, but given the low number of participants in this age group causing wide confidence intervals, and that this study was not powered to fully evaluate subgroups, this should be interpreted with caution. When looking at the local reactogenicity population, there were more local reactions in the treatment group than the placebo group. However, most were grade 1 to 2 in severity. The most common solicited reaction was pain, and the incidence was comparable between doses. Grade 3 local reactions were more common after the second dose, and the driver of this was injection site pain. The majority of reactions occurred on the day of vaccination or the day after and generally persisted for about 3 days. There were a few local reactions that persisted beyond 7 days in the treatment arm, but not in the placebo arm, and these were mainly induration, erythema, and pruritus. In a subgroup analysis of adults greater than or equal to 65 years of age or older, rates of solicited reactions and other unsolicited adverse events were comparable to all other participants. Systemic reactogenicity outcomes again were higher in the treatment group than in the placebo group. Overall, solicited systemic events increased after dose 2, with about 55% reporting after dose 1 and 79% reporting following dose 2. Overall, the most noted systemic reactions were headache, fatigue, and muscle pain. Most of the systemic reactions were grade 1 or 2 in severity. The most common grade 3 reactions occurred after the second injection and were fatigue, myalgia, headache, and arthralgia. It is important to note that systemic reactions were observed with a median onset of 1 to 2 days following vaccination and resolved within a median duration of 1 day for both the younger and older age groups. For overall adverse events, data was available for 28 days post any vaccination in 30,351 participants and are shown here. Unsolicited severe and serious adverse events were similar between treatment arm and placebo. The most commonly reported unsolicited adverse event after injection was headache. Serious adverse events, which were defined as adverse events that prevented the participant's daily activity, that had a greater frequency in the treatment arm regardless of age group, included those related to local and general reactogenicity reactions, such as injection site pain and headache. There were 13 deaths as of December 3rd, 6 in the treatment arm and 7 in placebo. None were considered to be related to vaccine or placebo, and only 1 was confirmed to be related to COVID-19, which was in the placebo arm. There were no anaphylactic or severe hypersensitivity reactions noted at the interim analysis, and there were 4 total reports of Bell's palsy, 3 in the treatment arm and 1 in the placebo arm. Currently, there are insufficient information to determine a causal relationship between reports of Bell's palsy and vaccination. In summary, based on current data from both preclinical and clinical trial, mRNA 1273 is safe and effective against COVID-19 with 94.1% vaccine efficacy. Serious adverse events were low and consistent between treatment and placebo arms, including in patients who were SARS-CoV-2 serologic positive and asymptomatic at baseline. Again, just as with BNT162b2, it is important to note that the systemic side effects were more frequent in the treatment arm, and this should be taken into consideration when vaccinating groups of people such as healthcare workers. Systemic symptoms can overlap with many of the currently utilized symptom screening tools for workplaces. Encouraging vaccination at the end of the work week may be appropriate to avoid this overlap. Similarly to BNT162b2, this large phase 3 study not only delivered positive results and was produced with large numbers in a short period of time, but also provides proof of concept for RNA-based protection against infectious diseases. In terms of what is missing from the current data, there are a few gaps that this leaves us with. The major gaps for the general population are the long-term safety and efficacy data, as well as the vaccine's efficacy against asymptomatic disease and protection against severe disease. This study was not designed to evaluate efficacy against asymptomatic disease and disease transmission. Although, due to the added serologic testing prior to dose 2, there was limited data on asymptomatic disease available, which numerically saw lower numbers in the treatment group. Again, since this study was not specifically designed to address this as an endpoint, a conclusion cannot be drawn. There were also too few cases of severe disease to be conclusive about any kind of overwhelming efficacy for prevention of severe disease. However, what is showcased in the interim phase 3 data is promising. Further data will be needed to address these issues, and until then, continued public health reminders such as continued mask wearing and social distancing is strongly encouraged. Further data is also needed in specifically excluded populations such as pregnancy, breastfeeding, and immunocompromised. There are currently no data available to speak to safety and efficacy in these populations, with the exception of the DART animal study, which showed no observed adverse effect at 100 microgram level in rats. As more data from clinical trials and patients receiving the vaccine via EUA access is available, we will hopefully have more information to speak to in these specific populations. Both Pfizer and Moderna's vaccine candidates were approved for emergency use authorization. There are a few key differences between these two vaccines that are important to know for healthcare workers and vaccine recipients. The Pfizer-BioNTech vaccine is 2 doses, 21 days apart, and for ages greater than or equal to 16 years old. It also must be stored at negative 80 degrees Celsius, and at room temperature is only good for no more than 2 hours undiluted. There are about 5 doses in the vial, although there are reports of many being able to have enough left in the vial for 6 doses and a sporadic 7th. It is important that you should not combine the residual doses from multiple vials to make 1 dose due to the lack of preservatives. This vaccine also is not interchangeable with other vaccines such as Moderna's. Of note, Pfizer's vaccine recently received conditional marketing authorization via the European Medicines Agency on December 21, 2020, for patients aged 16 and older under the name Comirnaty. For Moderna's vaccine, it requires 2 doses 28 days apart and is approved for ages 18 and older. It should be stored at negative 25 degrees Celsius and is good for up to 6 hours once punctured. There are 10 doses available per vial, and it has no preservatives and is not interchangeable with other vaccines. Moderna's vaccine is currently under European Medicines Agency evaluation for conditional marketing authorization. When comparing adverse events between the 2 vaccines, local and systemic reactions are similar between vaccines with numerically higher frequency in the Moderna vaccine candidate mRNA 1273. Values shown are specifically for solicited reactions following the second injection. Other pertinent severe side effects between the 2 vaccines are rates of Bell's palsy and anaphylaxis. Although currently Pfizer carries the bulk of the concern with anaphylaxis, since there was 1 occurrence in the clinical trials and 6 out of 272,000 doses given following emergency authorization. Moderna is being watched closely for any anaphylaxis occurrences during emergency use. The rates of Bell's palsy during both Moderna's and Pfizer's clinical trials are consistent with background rates in the population. However, given the numerical imbalance between treatment and placebo arms, this event is also being followed closely during emergency use. As noted previously, there are limited to no data regarding safety and efficacy of these vaccines in patients who are pregnant or breastfeeding. Currently, Moderna has its developmental and reproductive toxicity study data available and saw no adverse effects at the 100 microgram dose level in pre- and postnatal development. During the clinical trials, there were 13 pregnancies reported in the Moderna trial and 23 in Pfizer's trial. 10 of the 13 from Moderna's trial were still ongoing, with 1 experiencing a spontaneous abortion at 7 weeks gestation, not considered to be related to treatment arm. 1 had an elective abortion at 6 weeks gestation, and 1 was lost to follow-up, and the pregnancy outcome is unknown. Pfizer's participants are still continuing to be followed, and outcomes will be available in the future. Additionally, both the American College of Obstetricians and Gynecologists and the CDC do not recommend against vaccination, but rather state risks versus benefits should be weighed given the risk associated with COVID-19 disease in pregnant patients. They also suggest having a conversation with your healthcare provider if pregnant or breastfeeding. They recommend that breastfeeding should not be avoided following SARS-CoV-2 vaccination. Specific resources to learn more about vaccine recommendations and frequently asked questions are listed here on this slide. The CDC provides excellent guidance for both healthcare providers and patients receiving vaccine, as well as separate information for both EUA-approved vaccines available. Thank you very much for watching. I hope this was a helpful summary of the SARS-CoV-2 mRNA vaccines. Again, this video will continue to be updated as information becomes available. If you have any questions regarding the material presented in this video, please reach out.